Thanks, Marion, and thanks to everybody for the invitation to speak today. Circadian rhythms, as Marion said, are these daily rhythms. Probably the most familiar to us is our own sleep-wake cycle. These are rhythms that are not unique to the animal world. They're found in many bacteria. They're also found in plants. And in fact, the first model organism for studying circadian rhythms almost 300 years ago was a plant, which was studied in France by a French scientist who had the idea of the leaves were moving in a daily fashion, and he had the idea of taking this plant into his basement and seeing if the leaf movements would continue under constant conditions in his basement, which he reported that they did. And we have a place in New Mexico, and I think I can show you what this looks like. This isn't the same plant, but it illustrates the same kind of phenomena. You can be walking in this semi-arid canyon land. This is northern New Mexico, and you run across these datura plants, and there's not much there during the day to look at. The leaves look fine, but these blooms look like they're all finished. But if you come back in the early evening, you see something that looks like this. The blossoms open up completely and they close at dawn. So during the day they're closed, and at night they're open. Now we have reasons to believe that this makes adaptive sense, because this plant is fertilized, these flowers are fertilized by a night flying moth, and you're in an arid environment. It takes a lot of water to pump that modified leaf open like that, and transpiration, loss of water will surely affect you in those arid environments. And so we believe this is an adaptation that simply says fertilization, pollination can only occur at night, produce a structure at night, withdraw it during the day. For decades, we've known from animal studies that these are endogenous rhythms. I mentioned this plant experiment, very old plant experiment, more recently, 1959, as you see in this figure legend. This is from a Cold Spring Harbor symposium on biological clocks in 1960. And as the figure legend says, this is a pattern of activity that is seen in a flying squirrel that has a running wheel in the cage. And each horizontal line is a, I'm not sure which of these, each of these horizontal lines is 24 hours. And the deflections off the line, these heavily banded regions, are times when the animal is on the running wheel. That's being recorded on an event recorder. And this is a 25-day-long experiment. There's no light-dark cycle or temperature cycles in this experiment. The animal was originally on a light-dark cycle, but released into constant darkness for this experiment. And you can set your watch by timing your getting on and off of that running wheel each day, maybe a 3 or 4 minute variation that's seen throughout the length of this experiment. And again, the fact that this is going on in the absence of any apparent environmental influences reinforces the notion that these are endogenous timers of some sort that create some 24-hour activity.
You got up earlier each day a little bit.
Yes, yeah. This is a good point, Jim, because circadian clocks are called circadian because they're about a day. They're not exactly 24 hours. And geologists or those interested in the history of the Earth will tell you that the Earth's rotation has changed over the last 4 billion years. And these clocks are loose adaptations to a changing periodicity of our Earth. And as you'll see a little later, it's quite easy to make changes in the principal molecular components of these clocks and get significant changes in the periodicity of these endogenous clocks, so they can follow different environmental periodicities. But because ordinarily the animal is exposed to an environmental cycle, and we have mechanisms that I'll talk about that allow them to entrain their behavior to the environment, it's sufficient that they have near 24-hour rhythmicity that is then adjusted in an environmental cycle to precisely 24 hours. And I'll say more about that, the mechanism of that later. There are other examples of the use of these clocks for non-24-hour behavior. And an example here is from von Frisch in the early 1900s, who was studying the dances of honeybees. And of course, honeybees live in a hive, but you have foragers that are going out in the field locating sources of food, and they have behaviors that they perform when the foragers come back to the hive. They move into the hive, they get on the wall of the hive in the dark, and perform these these locomotor motions that are interpreted by other foragers in the hive and give signals with regard to where the food source is. So one of these, again, this is all from von Frisch, about 100 years ago. One of these is called the round dance, and you see the trajectory that this animal takes on the wall of the hive. This simply means there's food close by, go out and get it. I'm not giving you much information about distance and direction. But there's a much more interesting dance that the foragers will perform if the food is at a greater distance. This is called a waggle dance. It's a figure-8 dance where the bee goes through this figure-8 over and over and over again. And when it goes through the middle of this figure-8, it waggles its abdomen. The frequency of waggling has information that indicates distance to the food source. Position of the food source is indicated by the direction of this crossover in the figure-8. So, for example, if you have a hive in a field, and you have a feeding place that is aligned with the direction of the sun from the hive, then this dance will be vertical on the inside of the hive. On the other hand, if the feeding place is offset from the direction of the sun, in this case by 40 degrees, the waggle dance changes by 40 degrees. So the animal produces a different behavior, a related behavior, but a different behavior that indicates the direction relative to the sun that any other forager should take when they move outside the hive. Of course, the problem is the sun moves, or at least we move, and the perception of the bee is that the sun's moving. And these dances, these waggle dances, go on for hours by the same forager on the inside of the hive. And what von Frisch learned is that over the course of those multi-hour dances, the figure-8 dance would shift its orientation in a fashion that accurately predicted where the sun was going to be. We now know from a variety of studies that they're using their circadian clocks to perform that kind of clock. So this is much more contemporary information. This is information about gene expression from an entire head of a fly. And I put it up here for a couple of reasons. This is about 150 genes that have been stacked one on top of the other. Each row is the expression of the gene over a 6-day interval. So down here at the bottom, a green box means a low level of RNA for that gene. A red box means a high level. And what you see are red and green stripes that alternate, 6 red and green stripes that alternate and run at a sort of diagonal across this. The flies were exposed to a light-dark cycle, 12 hours of light, 12 hours of dark, and then 24 hours of constant darkness, and that was repeated 3 times for 6 days total. And what has been done here then is each gene has been stacked within this illustration in a way that the nearest neighbors are closest together, so that you have this continuous picture. And this by no means is all of the genes that are oscillating within the head. There are at least 300 that are oscillating in the head, but part of the problem is these are whole heads. You've got different parts of the brain, you've got the antennae, the eyes, all mixed together, and only the most robust of these rhythms will be there for observation. But an important point that I wanted to make with this is that we have, I've told you about oscillating day-night behavior, and clearly these clocks are not needed just to inform the organism about 2 alternating states states of the environment. Is the sun up or is the sun down? This is much more like a music box where you have a sequencing of gene activities, so that by knowing the particular amplitude of a given gene, you can predict what time of day it is, and presumably this is in order to get groups of genes to work together in some coordinated way when they have a job to do at a specific time of day. So it's not a pendulum that swings back and forth, but a series of outputs through these gene expression programs. Now this is similar to a fusion of what I just showed you, plus 3 days that are measured in 3 mutants that we've studied over the years. I'll tell you the names of these in a moment, but these are null mutations that knock out the clock with regard to behavior as we assess it, and also, as you see here, also knock out all evidence of the rhythmicity of these genes that in wild type and normal flies was previously so rhythmic. An important note on this is that these 3 days are light dark cycles. So these RNAs were collected in the presence of a light dark cycle, and yet the rhythms are gone. So presumably when these clocks first emerged, they were responding to environmental signals and were very closely coupled to them. They're still very closely coupled to them, but we are now so deeply invested in the clock mechanisms themselves that they will no longer, the system will no longer respond to just environmental cycles. The environmental cycle has to be interpreted by these endogenous clocks in order to get these gene expression patterns to become rhythmic. So the very first of these genes was picked up by Ron Kanaka and Seymour Benzer working at Caltech in the early 1970s. And the second gene in the flies was found by our group by using a method that Kanaka used. Kanaka found his mutants in the first 200 flies. We went through 7,000 bottles of flies to find the second mutant. And so there was a considerable lag between the finding of the first mutant and the finding of the second mutant. The assay that was used here is a hatching rhythm. The flies have cocoons, essentially pupae that they form, and the adult hatches out of those pupal cases in the early morning, and so you can see rhythms within a population hatching that is dependent on these circadian clocks, and so mutants will either abolish those rhythms or speed them or slow them down. And another way of, a more contemporary way of looking at this, the way we do this now, is just to monitor individual flies. So we will mutagenize flies, we will feed them a chemical mutagen that causes chromosomal breaks, and then we will put the progeny of those flies in individual flies into these cuvettes, which slide down into a channel where we have a light-emitting diode and a photo transistor. So we have infrared light shining across this cuvette, some fly food in the cuvette and a single fly, and when the fly moves back and forth, it will break that beam, and we've got a simple computer system and software that will give us products that look like this. So these are all mutations of a single gene that we call double time. The reason we called it double time is the first people we picked up had a fast rhythm. It had an 18-hour rhythm instead of nearly 24-hour rhythm. What this demonstrates is what these activities look like. So we've got 48 hours plotted here. The animals are initially exposed to a dark light cycle and then put into constant darkness. And thereafter you see these are marks where the fly is moving. And so you can see the fly is moving and resting rhythmically. And as Jim pointed out earlier in wild type, this is a mutant, but the animals are working up earlier and earlier each day and beginning rest earlier and earlier each day. So this is one version of a mutation. It's actually a point mutation that affects the structure of the protein, but a different mutation at a different part of the same protein produces a very different phenotype. The phenotype is a long period instead of a short period. And so far, it seems like we can get this collection of long period and short period mutations that most, if not all, of the genes that compose this clock and generally loss of the gene, if it doesn't produce lethality, produces arrhythmicity. So I want to tell you a little bit about two genes that work at the center of this mechanism to produce molecular oscillations, not only their own oscillations, but oscillations elsewhere in the system. And these two genes are called period and timeless. And both of these genes are expressed with a circadian rhythm, that is in a light-dark cycle or even in constant darkness. They cycle in their abundance, their transcripted protein abundance. And importantly, unlike the genes that are all over the map that I showed you previously, the rhythms are tightly coupled, so they always go up and down together. A mutation in one of these genes will affect the periodicity, the rhythmicity of the other, so that, for example, if we have a mutation that causes a short period behavior and short period oscillation of the period gene, we'll get short period oscillations in the timeless gene as well. So both of these genes are tightly coupled in their function within this clock. Leslie Vosshall, a graduate student in the lab, a number of years ago, found another interaction between these two genes that we feel is very important. This is the optic lobe of the fly. The brain would be down here, the central brain. These are the photoreceptors that make up the compound eye of the fly, and these dark-colored oblong structures are the nuclei of these elongated photoreceptor cells. So in the middle of the night, the period protein is found in nuclei. In the middle of the day, you don't see the period protein, and you'll see one later. This is what we would see in a wild-type fly, but in the timeless null mutation, the other partner gene, Leslie never saw any accumulation of this protein. So somehow the timeless protein is required for the accumulation of the period protein. What Leslie did was to make a fusion protein, a gene that would encode a protein that was part period protein and part beta-galactosidase, bacterial. Maybe this is some microbiology connection. But a fusion protein that's part bacterial, part Drosophila, and that protein is unusually stable, and so now she can detect this protein. These are normal heads from normal flies, and you see this nuclear association of this fusion protein, this period fusion protein. These are from a timeless mutant. They can still make the protein, but the protein gets stuck in the cytoplasm of these long photoreceptors. It doesn't make it into the nuclei. So this brings us to a model, which I've only told you the barest parts of, but I'll just jump ahead to show you how we think this system works. So we've been talking about period and timeless. We have two genes, period and timeless, that are, like all genes, reside in the nucleus. They produce RNA that is exported to the cytoplasm for instruction to build protein all day long. And that production of RNA is dependent on two activator proteins called C and B, clock and cycle. So all day long, clock and cycle are deriving expression of RNA off of the period timeless genes. All day long, those RNAs are being translated into period and timeless proteins, but as I'll explain in a minute, no period or timeless protein accumulates all day long. In order for either of these proteins to get back into the nucleus, they have to pair. They physically associate with one another. And as I'll tell you, following some additional steps, these period and timeless dimers will move back into the nucleus. And the important feature here is that they physically contact the C and D proteins and repress their activity, the net effect being that they will shut down the further transcription of period and timeless gene process. So, as I mentioned, all day long, we're not accumulating the period and timeless proteins, and that's because we have two breaks in the system. One is called double time, which is a kinase. Kinases add phosphate groups to proteins and often mark them as targets for other proteins. In this case, double time phosphorylates period, which then causes it to be rapidly broken down. And for timeless, the break is a protein called cryptochrome, which is a protein called a photolyase, which is activated by light. In the presence of daylight, cryptochrome is activated and binds to timeless. That recruits other proteins that modify timeless and cause its destruction. So all day long, light is causing the destruction of timeless. If timeless is not around, you cannot form these period timeless dimers, and double time will cause the destruction of per. When the lights go off, cryptochrome will no longer be activated. You'll be able to make these complexes. They'll be able to get into the nuclei and close this feedback loop. So we see, and it's easy to see how this mechanism cycles in the presence of an environmental shift from day to night. Of course, the question that we all want to know is what makes this thing cycle when there is an environmental cycle, and that's what is meant by a circadian rhythm. These are endogenous clocks we're interested in. So a few years ago, a student in the lab, Pablo Mayer, who his background actually was in physics. He'd had no biology coming into the lab, but he wanted to do something that was a little bit different from what was going on in the lab before him. And Pablo decided that he would try to build a part of a clock in some naive cultured cells that were derived years and years ago from embryonic Drosophila. So what Pablo did was to make two fusion proteins that had fluorescent tags on them. One is for the period protein that had a blue fluorescent tag on it, and the other was for the timeless protein with a yellow fluorescent tag. So these images are of the same cell at the same time, but in this case you're looking at the distribution with the white is the distribution of the per protein, and then this the distribution of the timeless protein. And I'm now going to show you a movie. These were live cells that were made to produce these proteins, and then the results followed in a looped movie to show the series of steps that these proteins undergo. So at the beginning of the movie, which is right now, you see first cytoplasmic localization of these proteins. Then the proteins come together in a series of puncta in the cytoplasm here. And then shortly thereafter, they both move rapidly into the nucleus. So Pablo had achieved a system in which he could artificially produce two of these proteins in this cultured cell system and get some of the movements that we would ordinarily see happening within the cells of the brain of the fly. And these are just steps in this process, breaking down the movie. Importantly, showing that these puncta that contain period, for example, are the same puncta that are also carrying timeless. So proteins are coming together in these puncta, and then after about 5 hours, there's a transition from these cytoplasmic associations, a transition to a nuclear association for these proteins. So among the questions we have is, we know that you need to have both of these proteins, and they need to physically interact to get into nuclei. So what Pablo wanted to know is, what part of this process, where did the physical interactions between these proteins align in this process? And we have an approach to asking this question that is called FRET, fluorescence resonance energy transfer, in which if you have two proteins that both contain fluorescent tags, shining light on one of those proteins, or shining light on the experiment that only one of those two proteins is excited by, can cause, if they're close together, close enough together, about 100 angstroms from one another, you'll get energy transfer from the excited protein to the protein that is not excited by that wavelength. So in this case, Pablo put light on the system that would excite the CFP, the blue fluorescent protein, and search for FRET, which was a 575 nanometer emission that would be appropriate for the YFP partner. And this is a movie that shows what he saw. So the top two figures are much like what I showed you before, where we first have cytoplasmic accumulation of the proteins, the puncta, and then nuclear accumulation. What we see down here is that FRET is high when it's red and yellow, low when it's blue, And Fred's high right at the beginning of the experiment, right after these proteins are produced, and stays high until the proteins move to nuclei. So in a way, this trajectory that we imagined is turned around a bit. It looks like the proteins can come together, but that's not a simple ticket to get into the nucleus. Something else is happening that takes another 5 or so hours before those two proteins can get into the nuclei. And when they do go into nuclei, they may go in separately, even though they've been staging in the cytoplasm together for about 5.5 hours. So in an effort to understand what it is that's holding these proteins in the cytoplasm, a postdoc in the lab has recently been looking at mutations that affect the period and timeless proteins, and more importantly, affect the timing of this process, both in those cultured cells and in the flies. These are pictures that come from a fly, or wild-type flies, normal flies, and also flies that have mutations in the timeless protein that are that affect target sites for adding phosphorylation by a kinase called GSK3. So we have two phosphorylation sites that we know are targets for this kinase in timeless. And what we're looking at here is the relative subcellular location of timeless and wild type at the same time that we're looking at localization in this mutant, where Dennis has removed two of the phosphorylation sites, substituted serines, which are phosphorylatable, with alanines, which are not. And PDF is a neuropeptide that simply labels these clock cells in the brain with green. You can see the nuclei are vacant with this marker. And what we see in the wild type is that at ZT22, which is 10 o'clock at night, 4 o'clock in the morning, 2 hours before dawn, we have the nuclei filled with timeless protein and the cytoplasm filled with this neuropeptide. If we look at the same time of day with this mutant TIM2A, we see donuts all the way down. And in fact, we continue to see donuts until we look much later. CT5 is 7 hours later than ZT22. What we've done is keep the lights out. The dawn never came. We just kept the lights off. 7 hours later, we finally see in the mutant the nuclear accumulation of this altered protein. And of course, in wild type, by that time, the protein is gone from the system. So this is a summary of many, many mutants of that sort. And what Dennis realized is that he can change the clock almost at will by modifying these phosphorylation sites. So for example, if he produces mimics of phosphorylation by converting serines to aspartates, which behave as if the protein's already phosphorylated, the residence time in the cytoplasm is only 2 hours. If he knocks out all of the phosphorylation sites, all 5 of them, he's got a 17-hour delay. This is a 35-hour clock. So this is a very adaptable system. You can make just a few mutations and change enormously the periodicity of this clock. But a very important component is this cytoplasmic regulation. Now, phosphorylation isn't the only thing that comes to bear on this process. I've been telling you about GSK3, is that one kinase. But we also know a second kinase, CK2, also promotes this nuclear accumulation through phosphorylations of yet other sites on timeless. Double time affects this process by phosphorylation sites, this time on PER. And OGT and OGA affect glycosylation. There are N-acetylglucosamines, sugar residues that are added to PER. And adding these to PER delays this process, and taking them off with OGA accelerates this process. So somehow, this whole series of events, this 5.5 hours, is a reflection of these combined activities, ensuring that, in a sense, there's an interval timer that holds the protein in the cytoplasm for the correct amount of time before releasing it into the nucleus, which of course affords an oscillation. So just to summarize this part of the first part of the talk on circadian clocks, Again, they're composed of a small group of conserved genes. We have these genes, and in fact, there are sleep disorders that have been linked to mutations in both period and double time. There are families, whole families, that have members that have a disorder called advanced sleep phase disorder, where they have difficulty staying awake past about 6 o'clock in the afternoon, and they're up running around at 4 o'clock in the morning. And they have mutations either in the kinase or in PER, the target of that kinase. So we know this, these genes are not only present in us, but they have the same role in controlling our circadian rhythms. The clocks run autonomously, as I just told you, but they can be entrained to environmental stimuli, and I gave you one example with light. I'll mention one other example because they're also entrained by food, and this is what jet lag is all about. If you have a mouse, and which is a nocturnal animal under most conditions, and you allow it to eat only in the middle of the day, it's going to have to wake up, eat, go back to sleep. It'll get back on its running wheel and run all night long, and it'll produce after several days a pattern in which the animal anticipates the delivery of food, will wake up an hour in advance, eat the food, go back to sleep. What happens in that animal after about a week is that if you look at tissue in the brain that controls the locomotor activity, and this is all in the light-dark cycle, the phase of activities, the oscillations in the brain have not changed. But if you look at the liver has been shifted by almost 180 degrees. So you have an animal that is utterly desynchronized. Its peripheral organs are running as if it's in Tokyo, and its central nervous system is running as if it's in New York. And I think something very much like this happens when we take a transatlantic flight or a flight from here to the Far East or back, for example. These molecular oscillations are generated by delayed feedback loops that I've described, and again, multi-hour delays are associated with regulation of these protein interactions, the stability of the individual proteins, and a control of subcellular location. And there are probably additional components to this regulation as well. So I'd now like to change the subject and talk about sleep as opposed to circadian rhythms. They're really two related but different pieces of the problem. And I want to also argue that what we're doing with flies may tell us something about mammalian sleep. So first, I've already shown you activity records where you see that, like us, they have consolidated intervals of rest and activity. They're diurnal, as we are. When the lights are on, they're active. When the lights are off, they're inactive. If we look at a resting fly during the day and compare its responsiveness to something like a puff of air at night, we find that the arousal threshold is very different. So rest during the day and a mobile fly during the day responds much more rapidly to a stimulus than an animal resting at night. There are circadian controls that I've already described, but there are also homeostatic controls. That means if we sleep deprive flies, just as if I sleep deprived you, you will try to catch up on that sleep on the next day, and I'll tell you how we assess that. There are age-related changes in these patterns. So our activities, our sleep-wake patterns erode as we age, and that happens in flies as well. There are conserved responses to drugs that promote sleep or wakefulness. An example here is modafinil. Another would be caffeine. The flies and we have similar responses. And there are also brain activity changes. You can't do EEGs on a fly, but you can measure something called the local field potential, which is you put a probe in the middle in the central brain, and you look for electrical activity that's coordinated with rest during the day or during the night, and what you find is that there are very different electrical activities corresponding to rest, depending on whether it occurs during the night or during the day. Yeah.
Is this something universal, or are there organisms, living organisms, that don't have circadian rhythms?
Most organisms that have been studied in detail you find some circadian marker that you can follow. The easiest way now are molecular markers that tell you, even in C. elegans, for example, where there were not obvious, robust circadian behaviors, we now know that there are molecular oscillations, circadian molecular oscillations that are very robust. So the trick has been finding the assay for the clock, much more so than it's not being. So the approach we've taken in this, and this is, I'll tell you about work that was done by a postdoc that's now got her own lab at Harvard, Dragana Ragulja. And what Dragana did was to take advantage of two centers, one in Japan and one in Vienna, where there are stocks of strains of flies. Each strain of flies makes a suppressor, a specialized suppressor that is made of nucleic acid, made of RNA, that when expressed in a fly, and particularly when expressed with a particular pattern within the anatomy of the fly, will seek a target and shut down that gene target. So this is a way of simply marching through the genome. There are 13,000 protein coding sequences in the fly. She marched through 4,000 of those, so you can go one by one, and you eventually know when you're done. And you can say, what does suppressing the activity of this gene do with regard to this phenotype, these sleep duration phenotypes that we want to look at. So her approach was also to, and the suppressor is called RNAi, and her approach was to selectively produce RNAi for each of these lines using a driver, a regulator, that causes it to be expressed, the suppressor only to be expressed in postmitotic cells of the nervous system, non-dividing cells of the nervous system. And that is called OLV, in case I slip into jargon. So this is, she found about 20, in screening 4,000, she found about 20 that had very strong effects on sleep duration. And this is one that caught her attention because it's a cell cycle gene. It's been studied deeply because of its involvement in another kind of cycle, the cell division cycle. And it's called a regulator of cyclin A1. And the RNAi suppressor to regulator of cyclin A1 causes flies to have a mean level of sleep that's about 35% lower than the average level of sleep that you see in a normal or a wild-type fly. So wild-type flies will sleep for about 900 minutes a night. There's a distribution. Each one of these dots is a different individual, a different normal individual, these are all different mutant individuals. But if we look in this group for an individual that has almost normal levels of sleep, we still see an important difference from the normal flies. And that is, normal flies show this consolidated segregation of activity and rest each day. Here we have a much more asynchronous pattern of rest and activity. So since regulator of cyclin A1 regulates another cell cycle gene called cyclin A, in the cell cycle, Dragana asked whether cyclin A was also a sleep-regulating gene, and she found that the answer was yes, it is. So again, using a suppressor this time for cyclin A, this time we've knocked sleep down almost 50% in these flies by knocking out this particular gene. If we look at the nature of the change in the sleep pattern in the fly, there are a few things we can assay. First is there a change in the amount of activity that the fly is producing per waking minute? And the mutant form is shown here in red, and there's really no significant change in what is seen from the wild type. But where we do see differences is in the length of time the flies spend sleeping or resting during the night, not as a over the entire night, but as bouts of sleep. So many of you are aware that you go to sleep, you wake up briefly, you fall back asleep, you wake up, you fall back asleep. And flies are very much that kind of a beast. And in fact, on the average, a bout of sleep in a fly lasts a little less than an hour, about 45 minutes. So flies will typically have a bout of sleep, wake up, and then fall back into sleep again. The cyclin A mutants have sleep bouts that are only about 20 minutes long, about half the length of a wild-type fly. And although there's a little more, there are a few more bouts of sleep every night in the mutant flies than in the normal flies, not nearly so much enough to make up for this deficit in the duration of the individual bouts. So the flies are able to initiate sleep, but they're not able to maintain it and wake up prematurely and then reinitiate again. This phenotype, you don't have to use this special suppressor. We can use mutants, which when present in 2 doses would kill the fly, but when present in 1 dose don't kill. And in those cases, what we see is that we have a suppression of sleep. And in fact, we can combine those mutants with this kind of a suppressor and get even more extreme losses of sleep that I won't go into. Another problem these flies have is that their first bout of sleep each night is grossly delayed. So when you turn out the lights, a normal fly will have its first bout of rest within about 40 minutes. Routinely, these mutant flies for cyclin A fall asleep for an hour and a half. So the first bout of sleep comes late, and then when they finally manage to have a bout of sleep, it's ended prematurely. Now, these are tests that have to do with rebound. Again, if we deprive the animal of sleep, what's its response on the following day? The way we do this is we have that rack of tubes that I showed you a few minutes ago on a cam, so that every minute or so it vibrates for a few seconds. And we can run this all night long or all day long, and we can look at the effects on the sleep pattern. We don't see any effects on the sleep pattern if we do that mechanical vibration during the day, but when we do it at night, we see profound effects on wild-type flies and also on these mutants. So in wild-type flies, doing this all night long cuts sleep or the duration of rest in half. So these are 2 examples of wild-type flies, normal flies, who are cut down to about 500 minutes of rest per night. And we also cut the mutant in half. So although they only got half as much sleep in the first place, they're now down to a quarter the normal amount of sleep. The difference, though, that's important is that when we look at the amount of sleep that's recovered in the following day, the wild-type flies will sleep for 30% longer than they would had they not been disturbed on the following day, during the day. Whereas the mutant flies will only pick up about 10% of the sleep they lost. So they can't recover sleep, and they have a very hard time maintaining it as well. Now this takes a toll on the flies because they have rather short lives. They live about half as long as a wild type as a normal fly. So these are the suppressed cyclin A flies, and these are these normal controls. All of this is distinct from the circadian system. So for example, we can look at the hatching rhythm that I spoke of previously, and you can see that the mutant flies in red and the wild-type flies in black are hatching at a daily rhythm of constant brightness for 5 or 6 days without distinguishing themselves from each other. And if we look in the brain at the pacemaker cells in the brain that control circadian locomotor rhythms, we again see, PER accumulating just after dawn and just before dawn with very low levels of PER in between. And we see the same kind of accumulation profile in the mutants as we do in the wild type. So this doesn't seem to require a change in the circadian clock of the fly. Nevertheless, these cells have an interesting relationship anatomically to the locations of the cells that control circadian rhythmicity. So the cells in green control circadian rhythmicity, and they're marked with an antibody to the period protein, which you see here, here, and here. And for example, in the dorsal brain, this is the optic lobe. This is the dorsal brain. If we look just in this region boxed with purple down here, you see that the cyclin A cells are surrounding this cluster of circadian cells. So we have this intermingling of the circadian and sleep duration controlling cells. And we see this in several locations throughout the brain. For example, here's a lateral and dorsal region of the brain that contains, again, the clock cells and the cyclin A expressing cells, the sleep cells, and the circadian cells are once again closely connected. Yet another way of looking at this is these are the cells, this is the optic lobe in this cartoon, the central brain. These are the cells that control the locomotor activity, circadian rhythm and locomotor activity. They produce PDF, this neuropeptide, made in circadian 24-hour pulses. And those pulses are seen throughout these processes that go to the dorsal brain. When we look at where those are where that projects to, it's projecting to processes that are coming off of these cyclin A cells. So they may be projecting to other per-expressing cells, other clock cells up there, but they're also potentially interacting with these cyclin A carrying cells. And in fact, if we ask whether or not the cyclin A cells carry a receptor that interacts with that neuropeptide, the answer is yes. All of the cyclin A cells carry a receptor for that neuropeptide. So we think there's a functional connection there. And consistent with that, if we, instead of expressing this throughout the brain, this suppressor of cyclin A throughout the brain, if we express it only in cells that make the receptor to that circadian neuropeptide, we find the same suppression of sleep using that control as we do in the more widely suppressed form of the animal. The last thing I wanted to show you is an experiment that takes advantage of the fact that Dragan has been looking at lots of genes that with lots of different anatomical locations. We thought it would be useful to know if we could come up with a general method that would say if we find genes that have an effect on the duration of sleep and and can now identify the cells in which those are expressed, can we skip the gene now and just go to those cells and ask what is more directly the role of those cells in regulating sleep? And so what Dragan has done here is she noticed that this group of cyclin A expressing cells, 7 cells that express cyclin A shown here in red, are identical to the, or at least overlap with exception of one, this group of cells that expresses a different neuropeptide called cortisone. The reason that's significant is that cortisone is only expressed in these cells, and we can steal the regulator on that gene, and we can specify, using that regulator, we can direct the expression of anything we want into just this group of cyclin A expressing cells, and we can monitor, we can ask what's the effect, the behavioral effects of modifying the activity of those cells. So what she has done is she has used that regulator to control an ion channel called TRPA1. This is a temperature-sensitive channel that allows cations to move into the cell. So at higher temperatures, 30 degrees centigrade, the channel is open, will allow cations to flow into the cell that will activate the cell. At 25 degrees, that channel is not active, and so you revert to the natural uninformed function of the cell. And what you see here, there's a slight change in temperature in control flies, a slight change in levels of sleep that come from just raising temperature, but a profound change in the amount of sleep that comes with moving these flies from 25 degrees to 30 degrees. So 25 degrees, this is the activity and rest pattern. This is the activity and rest pattern at 30 degrees. And this is reversible, so that we can move these flies back down to this condition by simply cooling back to 25 degrees. So they certainly become much less active when we activate this one group of cells. So we can imagine going around the brain with any of these genes that we identify and associate anatomy with specific functions. So I'll just summarize this second half. We found this link between genes that control cell division and genes that are controlling sleep duration. There seem to be interactions between circadian cells and sleep cells. The cells are intermingled. The circadian cells are projecting neurons. a cyclone A expressing neurons, and they have functional receptors and neuropeptides that seem to interact. And finally, I just showed you the electrical activity of some of these cyclone A expressing cells can be modified in ways that affects the level of sleep in just one of the subcellular, in one of the subregions of the brain in which we find it. So I'll just point out again who did this work. Amit Patel, who's now at Penn, Leslie Vossall, who's a professor at Rockefeller, Michael Myers and Jeff Price all worked on those initial stages of the way the system requires period and timeless to produce that feedback loop. Brian Kloss and Justin Blau did a great deal of the work on the kinases that I spoke about. Pablo Meyer and Lino Saez developed the cellular system in which we can express these proteins and follow their movements and compare them to their movements in normal flies. And Dragan Rabilja did all the second half of this on sleep that I spoke about. So thanks again for inviting me to come and